Hey y'all, thanks so much for coming. And sorry, I'm way in the background. As I was telling Cheryl at the beginning, we're both in my office, and so I've given Jordan the reins to the big computer, which hopefully will not let us down. But again, thanks for coming. Jordan is a new PhD student in my lab, and as you know, based on her giving this journal club presentation, she's working with Project One, but also with Project 11 as well. And so she's wearing two hats and doing a great job. So she will be presenting the very first journal article. But before we get into that, a couple of things. Please, if you have any questions, you can put them in the chat, or you can raise your hand, use the raise your hand function. And I mean, we ask that you wait to the end, but if there's something like incredibly burning that doesn't make sense, please feel free to stop us during stop Jordan during the time. And the majority of this meeting is going to be the journal club, but we reserved 15 minutes or so at the end for Hector to go over how to search and sort through the database as well. So we do want to leave some time for that. So if time does get a little bit short, I may ask that questions be sent later. But hopefully there's a lot of great discussion. Don't hold back with questions. So with that, Jordan, you can share your screen and get started. Oh, is the screen shared? Let's see, let's go to here, and you can go to share screen, go to here and hit share, and then we'll go. And the title of her article is Cannibalism as a Possible Entry Route for Opportunistic Pathogenic Bacteria to Insect Hosts, exemplified by Pseudomonas aeruginosa, a pathogen of the giant mealworm, Zophobus moreo. And there you go.
Okay, so I'll start with a brief outline of the presentation we'll be giving today. And the first is introduction and background of the paper. The second, we'll go through the materials and methods. Then we'll go through the results, discussion, conclusion, and applications and potential future directions of research.
Sorry, guys. Okay, well, maybe you should just
click. Okay, so to start off with our introduction and background, the goals of insect mass rearing are to obtain large amounts of usable individuals. This could be for food and feed, it could be for scientific study. And the challenges of these goals are raising in a high population density, raising in high humidity, temperature concerns, inconsistent diet, inbreeding, and all of the above can result in stress in the insects. And stress leads to opportunistic pathogens, low yield, low productivity, microbial contamination, use case dependent, and then all of these problems cost the industry money. So talking about Pseudomonas aeruginosa, which is the main bacteria presented in this paper, where does this bacteria come from? It is ubiquitous in the soil and in fresh water. And opportunistic pathogens are pathogens that are just normal bacteria that can become pathogenic under favorable conditions. This can be stress, low insect immune system, and several other factors. So a little bit more about Pseudomonas aeruginosa being prevalent in the environment. It thrives in high humidity and high temperature. It's also highly sensitive to desiccation. And this high humidity is essential for the rearing in black soldier fly. It's also essential in the rearing of mealworm and several other insect species. So for Pseudomonas can form biofilms, and biofilms are groups of bacteria, and they increase protections for the bacteria that make up the biofilm. A biofilm can be made of a single bacterial species or multiple bacterial species working together, and they can be very difficult to eliminate. So on the slide here, I have a picture of a scanning electron micrograph of bacteria in a biofilm surrounded by a matrix. And so here is just a brief overview of how a biofilm is formed. We first have bacteria in a planktonic state that can attach to a surface. This could be pans that insects are raised in. This could be equipment. This could be all kinds of things. So we have formation of a monolayer and the production of a matrix. That's that yellow area around the bacteria. Then a microcolony forms before forming a mature biofilm. And this mature biofilm can break off pieces, detach, and revert to a planktonic growth. And this starts a new cycle. So just a little bit of bacteria left behind when trying to clean something up can result in a biofilm forming later. So one of the reasons that biofilms can be difficult to eliminate is, as you can see, we have our disinfectant sprayed here. Most disinfectants work by being able to contact the bacteria, and the bacteria on the inside of the biofilm are going to have more protection. So this is one of the benefits of a bacteria forming a biofilm. So moving on to the materials and methods of this paper, the first section, 2.1, was Pseudomonas aeruginosa isolation, identification, and preservation. So this was an industry experiment that was done. Dead and symptomatic larvae were used for this experiment that came from a producer. And the larvae were surface sterilized using an ethanol, water, and chlorine assay. The hemolymph was isolated and streaked onto a nutrient agar, and a predominant type of colony was detected, and only one type was detected on some plates. This single colony selected growth was also grown in liquid culture. It was gram stained and biochemical tests were performed. The DNA was also isolated with a commercial Qiagen DNeasy kit, and then PCR, and then gel band purification. The sequencing of PCR products was done using Blastin software. Some of the pictures I have on the right-hand side of the screen are free NCBI resources for blasting nucleotides or blasting proteins. So for section 2.2 of materials and methods, this is talking about how the mealworms were raised. The larvae were purchased from a local retailer and then placed in acrylic boxes for pupation. They were fed 1 carrot slice every 4 days using a diet developed in that lab. The details are listed below. They were hatched and randomly selected for mating, and within the new generation, the larvae were selected for OS injection, also called force feeding, and subdermal injection. The larvae were fed every 3rd day with a cube of agar and a bacteriological agar. All of the experiments were incubated in darkness at 28 degrees Celsius with an average relative humidity of 70%. So there were 4 different types of infection experiments that were done, and we're going to go over the materials and methods for all of them first. The first one was force feeding, also referred to as OS injection. The larvae were immobilized, and then a manual injection of 10 microliters of bacteria suspension were injected through the mouth to the larva. And then 2 doses of bacteria and 1 control were used, a high dose, a low dose, and then a negative control. The bioassays were performed in triplicate, and then the mortality data was recorded. For the second infection experiment, this was group exposure, and the group was exposed via the feed. So larvae were placed in a petri dish and exposed as a group to a concentration of Pseudomonas. The bacterial suspension was deposited on a feeding arena made with a carrot disc that was placed on top of a agar disc in the middle of the petri dish. And we've got a picture of that labeled figure 1 on the right-hand side of the screen. The larvae sample size was 6, and this was the max amount of larvae that could fit in these petri dishes. And this was representative of a high population density, as is typically used in commercial growing. The mortality was recorded over a period of 15 days, and the dead were not removed. This also mimics densely populated mass rearing conditions. And then whether the bugs were visibly cannibalized or not was either noted as yes or no. Missing larvae was assumed to be to cannibalism, and the authors defined cannibalism as visible signs of them being munched on, so to speak, or attacked by another larva. The group exposure experiment was also performed in triplicate. Okay, the next one was the number 3, the subdermal injection. So a selection of 20 larvae were injected on the ventral side within the 2nd sternite. A manual injection of 10 microliters of bacteria, and then 2 doses and a control were used, a high dose, a low dose, and then a negative control. Bioassays, again, performed in triplicate, and the mortality was recorded over a period of 5 days. The last infection experiment is called purposeful wounding, and it's wounding the larvae on purpose. So the wounding was done by removing the left hind leg tarsus of each of the 20 larvae with a pair of sterile dissection scissors. and 1 dose and 1 control was used. The dose and control was placed, or a control was placed at the bottom of the Petri dish, as in the picture on the right-hand side of the screen, just in like a little bubble in the middle. And then the purposely wounded larvae were placed in the wound, their wound was placed on top of the droplet. The bioassays were performed in duplicate, and then the mortality was recorded over a period of 10 days. Okay, going over the results. The first section of results reported were the observations on disease symptoms of the larvae. And so the disease larvae that came from the insect producer, the symptoms were noted were ceased feeding. There was a ventral curvature, meaning that they curved inward, sort of like doing a setup. And this happened over a period of 2 to 4 days, and then almost entirely lost their mobility. An insect body also became flaccid shortly before the insect died. And then the molting frequency was also reduced in diseased larvae. Cannibalism of the diseased larvae often occurred in the leg region, which was the reason for the location in the purposeful wounding experiment. Further results from this are melanization, which is dark staining. It starts with a small stain. In the picture labeled A, at the tip of the white arrow, you can see a black dot. And this staining increased over a 72-hour period. And melanization typically spread to most of the body in moribund larvae. This is indicative of septicemia, lysis of larval internal organs or tissues. The dead larva turned black at the proximal and distal ends. There was an unpleasant odor reported in the moribund and dead larvae. If the melanization was absent, they found the gut remained undisrupted, and melanization starting and spreading was indicative of gut disintegration. So the results for characterizing the bacteria Pseudomonas aeruginosa, bacilliform non-sporulated bacteria were found in the hemolymph, and this was done through in vitro cultivation and through microscopy. The in vitro cultivation resulted in 1 type of colony phenotype. They were green, described as convex, flattened as the growth progressed with round edges, and this suggests a monospecific infection. The in vitro liquid cultivation resulted in a yellow-green pigmented culture that changed color from green to blue when agitated. So the results of the characterization of Pseudomonas, there were several biochemical results to be compared. So in table 2, on the left-hand side, we have the isolate found in the larvae, and then the other biochemical assays performed out to the right. And then we also have compared to Pseudomonas aeruginosa and Pseudomonas fluorescens. And so you can see different tests here, such as the presence of gelatinase, glucose fermentation, and how these compared. The molecular identification, the length of the amplified 16S RNA gene fragment for the bacterial isolate was 770 base pairs. 95 out of the 100 sequences had significant alignments with Pseudomonas aeruginosa. The query cover was 100%, as well as sequence identity. For the infection experiments, the mortality rate of larvae for the first one, which was force-fed or OS injection with Pseudomonas aeruginosa, is listed here. So on the graph on the left side, we have a control, D1, which was the high dose, and D2, which is the low dose, and the mortality in percentages located along the y-axis. So we can see here that the high dose had a mortality rate of 59.6%, and the low dose had a mortality rate of 30%. On the right-hand side of figure 4, we have the cannibalism rate among larvae in the groups that were exposed to Pseudomonas. For exposure, the cannibalism rate was 56.6%, and then in the unexposed, the cannibalism rate was 33.3%. So the authors are still reporting seeing cannibalism in larvae that were not infected with Pseudomonas. So for the next round of infection experiments was group exposure, and that's where the larvae were exposed to Pseudomonas in the feed, and that resulted in no mortality. The cannibalism rate was compared in larvae exposed and non-exposed to Pseudomonas. In the larvae exposed groups, the exposed group's cannibalism was 56.6%. The unexposed group to cannibalism was 33.3%. The pictures over here on the right-hand side in figure A, this is a picture of the larvae molting in the group exposure experiment. And then in B, we have the injury due to cannibalism. At the white arrow, this is proleg, which is where the leg has been bitten off. And then at the yellow arrow, we have a picture of a dead larvae. The results for the infection experiment for the subdermal injection, the mortality rate is written here, but also in the graph. We had a high dose, low dose, and then a negative control. For the high dose, the mortality percentage was 98.3%, but significantly lower with the low dose for 41.3%, and the negative control had 0%. For the infection experiment with wound exposure to Pseudomonas, this was the purposeful wounding where the leg was snipped off and then placed in a bead of Pseudomonas. The wound exposure did not directly cause mortality, but the mortality rate of wounded larvae exposed for the exposure was 32.5%, and the non-exposure was 0%. So some discussion of the results that were presented were bacteria isolated from the larva was identified as Pseudomonas aeruginosa. This was done through biochemical characteristics and molecular identification. The disease larva exhibited symptoms consistent with Pseudomonas aeruginosa infection. That was weakness, sluggishness, cease of feeding, melanization. Again, that's that dark staining that's shown in the picture labeled A putrid odor, and then death. The first symptoms often appear unnoticed, and then the Pseudomonas aeruginosa is normally found in small numbers, but Pseudomonas aeruginosa can cause disease if it enters through the insect hemicoel. The wide range of Pseudomonas virulence factors, these can be pili, flagella, adhesins. So a picture of a bacterial cell here, the pili are labeled, and then the fimbriae are labeled here. And the flagella, which is important for bacterial locomotion, can also be an immune modulator. So the mechanism of Pseudomonas aeruginosa infection through the hemolymph is well understood, but the mechanism of bypassing gut defenses are poorly understood. There's a lot of defenses that the host has to kind of keep infections at bay from entering into the body cavity. This would be the microbiome, intestinal villi, and then gulp patches. These serve as an immunological organ in the gut. They're little patches of gut-associated lymphoid tissue. And then our subdermal infection mortality rates are consistent with other insects. Force feeding had reduced mortality as compared to subdermal injection. And the larvae force-fed with high concentrations of Pseudomonas had a high mortality after 24 hours post-infection. So high concentration led to allowing bacteria to pass through midgut epithelium to the hemicoel. This is something that was posed as a possible mechanism, but is in need of further research. The disease pathology is also dose dependent, as the research shows that higher doses typically had different pathologies or higher mortality rates. The opportunistic bacterial pathogens can affect insects reared in high population densities. The cannibalism rate was 23% higher for groups exposed to Pseudomonas compared to the non-exposed. I've got the graph for that shown again on the right. Cannibalism is reported to be a common behavior in mealworms by the authors, and then cannibalism affects individuals that are smaller, weaker, more vulnerable, especially at high population densities where food and space competition are more likely to occur. And potential solutions for that could be different types of insect or animal husbandry. Molting and pupation are susceptible stages for cannibalism. And again, this could be an insect rearing solution. The conclusions presented in the paper were that Pseudomonas aeruginosa is lethal to the mealworms injected intracholemically, force-fed, and wounding, but not the group exposure from feed. The mortality was higher for wounded larvae than non-wounded larvae, and that Pseudomonas aeruginosa transmission was enhanced by cannibalism. Contact with high doses of Pseudomonas should be avoided. It's reported as a workplace hazard, as it is also an opportunistic infection in humans. And then moribund and dead insects should be surveyed and removed. Insect feed and equipment should be inspected and assessed for bacterial contamination. And then cleaning procedures should consider biofilm formation. So the applications and future directions of this research, or some things that, kind of came to mind, would be, is it possible to filter insects by size via sifting trays to kind of reduce cannibalism? Will insects self-sort live and dead when presented with a new feeding arena in a new tray? Would that be a good way to kind of sort out the live larvae from dead larvae? Is it possible to remove insects with melanization? That could be an automation or an engineering solution. Is it possible to remove the bacterial load or reduce bacterial load by using light therapy? Is it possible to shift bacterial load by fermentation? And that changes the profile of how many and what type of bacteria that we're seeing in the feed. Do other species of insects have similar issues? And could increasing the insect immune system stave off opportunistic infections? Could the disease or loss of insects be an alternative source of revenue? Could insect probiotics confer protection to insects from opportunistic infection? That's something that we're already working on. And then additionally, does exposure confer resistance and immunity to future infections? So these are just some kind of research ideas that come off of this paper. And if you guys have any questions or want me to revisit anything, I'd be happy to do that at this time.